Avery, MacLeod, and McCarthy were essential scientists in the discovery of transformation genetics. These scientists asked the question, what is the chemical nature of the transforming substance? And determined that DNA is the transforming principle. Today we're going to dive into the experiment that they conducted in order to figure this out. First, we'll go into some of the background of where their ideas stem from. So the ideas in Avery, MacLeod, and McCarthy's experiment originated from the findings of Griffith. In 1928, Griffith conducted an experiment that demonstrated the idea of transformation within a substance. He used virulent and non-virulent strains of bacteria as well as heat to come to the conclusion that his bacteria was being genetically transformed during his experiment. But the big question was, what was being transformed? Avery, MacLeod, and McCarthy took Griffith's findings that substances could be genetically transformed and asked this big question. The experiment conducted aimed to isolate DNA, RNA, and proteins in order to determine which one caused transformation. First, they took a type S substance, which was virulent, and heated it to kill off everything and leave bacteria filtrate. They then divided this filtrate into 3 parts and added treatments to each one. The first one contained a treatment of RNase, which destroyed RNA. The second contained a treatment of protease, which destroyed protein, and the third contained a treatment of DNA, which destroyed DNA. They then mixed each sample into a type R bacteria substance. This substance was non-virulent. We see that these 2 samples containing RNase and protease produce type S virulent bacteria. However, the sample that contains DNase did not. So what do these 2 have in common? Both RNase and protease both still contain DNA, whereas DNase does not contain DNA. From this, we can determine that DNA is the transforming principle in transformation because it's the only one that still allowed transformation within the substance. This experiment provided further insight into transformation genetics and how substances could change genetically